So welcome back to the Classics of Immunology Journal Club. And before we get started, I want to remind you just to like the video on the YouTube site and click the bell to get more, to get notified. Scroll down and check out the website, my website, kendallasmith.com. Click on the writing tab and you'll find all the papers, including the one I'm going to discuss today. Actually, there's two papers that are on the website that deal with exactly the same thing. They were published in October of 1995 in Science, back-to-back papers. And I'm going to discuss the second one by a fellow by the name of Michael Karin, who is the senior author. The authors are Natalie Oplan, Joseph Donato, Caridad Rosette, Arno Helmberg, and Michael Karin. And this is coming from the University of California, San Diego. And it's entitled The Immunosuppression by Glucocorticoids, Inhibition of NFkB Activity through Induction of IkB Synthesis, the inhibitor of NFkB. So, and as I said, that's Science 1995. Now, this is another paper that really illustrates why it's important if you're a student of immunology to really delve into the history of all the things that have happened in the field over the last couple of hundred years. And it's really it's only about 100 years, so it's not a lot of literature that you have to deal with necessarily. But this goes back to 1960, 61, when actually even before that, in the 50s, glucocorticoids or steroid hormones were found to be immunosuppressive, and it came to be used in the clinic for the treatment of allergy and inflammatory diseases, as well as they seem to also work on leukemic and lymphomatous cells. So they were used as chemotherapeutic agents. 1960, the beginning of cellular immunology, began with Peter Noel, and we reviewed his 1960 paper where he described the use of phytohemagglutinin, or PHA, to activate the proliferation of peripheral human cells, human lymphocytes, which came to be found by 1970 as T cells. Well, the very first experiment after he figured this out, or after he discovered this PHA effect, what did he do? Well, he threw in glucocorticoid hormones into his cultures and showed that they inhibited this proliferative response activated by PHA. And he published that paper as a single author, as he did the first paper. Just a young investigator. He was an assistant professor at this stage. He was a cytogeneticist. He published that second paper in 1961 on the effect of prednisolone on PHA-stimulated lymphocyte proliferation. So this goes back to 1960. So we're talking 60 plus years here in terms of where we are now in 2022. Nothing really happened with regard to understanding what this glucocorticoid effect was. And everybody already knew that they were immunosuppressive and anti-inflammatory and anti-leukemic, so who cared how they worked, whatever. It wasn't until 1979 when my laboratory showed that once we realized and discovered that the stimulus for the proliferation was not the initial PHA that had been added to the cultures, but rather PHA stimulated the cells to release interleukin-2 and to express interleukin-2 receptors, and then they would proliferate in an IL-2 dependent fashion. So once we understood that, the very first thing we did was throw in some glucocorticoids into there to see what they would do. And what we found was they exerted a profound effect on the production of interleukin-2 that we could measure in our interleukin-2 bioassay with the CTLL cells, the long-term cultured cytotoxic T lymphocytes. They also inhibited the activity of IL-2, but it was only like 25 or 30%. So by far the profound influence was on IL-2 production. And of course, this is before we realized, before we knew that IL-2 at the protein level or at the activity level really was being coded for in the nucleus through a gene that encoded the interleukin-2 sequence. Excuse me, I'm hiccuping. So that was 10 years after. Oh no, that was 20 years after after Peter Noel published his first papers. So then again, nothing really much happened from 1979-80 until 1990, when several groups simultaneously showed that perhaps the mechanism whereby the glucocorticoids had this effect was on one of the transcription factors that had been shown to be important for IL-2 gene expression and production. And in particular, AP1 was thought to be involved. And there were several papers that, this is 1990 now, that showed that what happened was that the glucocorticoid receptor, which is in the cytoplasm, would translocate to the nucleus and bind to the AP1 protein, and somehow interfering with its interaction with DNA, and therefore it could not activate the IL-2 gene. Then in 1990, well, between 1990 and 1995, there were several papers out showing that there were several transcription factors involved in IL-2 gene expression. But it wasn't until Ellen Rothenberg from Caltech put it all together, really beautifully done experiments that she published in a couple of 3 papers in 1994 and 5, showing that there was a huge complex of transcription factors that would sort of glom onto the promoter region of the IL-2 gene, and that there were 3 factors in particular that were induced by the T-cell antigen receptor. One being AP1, another one being the nuclear factor of activated T cells, or abbreviated NFAT, and the other one was NFkB. Now, NFkB, which is what this paper that we're going to review today is all about, is a very important transcription factor in immunology. It's involved in many, many, many processes that are important for the immune activation and the immune response. So that's about where we sat until these 2 papers in Science in 1995. And this paper by Michael Karin's group at UCSD goes in the introduction. He makes the point that glucocorticoids are known to be very potent immunosuppressive and anti-inflammatory hormones. One of the things I didn't say before in terms of the background is it's sort of curious that there's this connection between the endocrine system, the adrenal cortical steroid pituitary axis, and the immune system. Why should that be? Well, it turns out that the immune system is not unique and separate and distinct from all the other systems. This is what the immunologists tried to sell. No, it's incorporated into the body, and it's regulated by, things like the endocrine system, among others. So glucocorticoids have a profound effect of regulating the immune reaction and the inflammatory response. The question was, what was the molecular mechanism whereby the glucocorticoids did this? And of course, they already knew that glucocorticoids, the hormones, were manufactured in the adrenal gland that sits on top of the kidneys, and floats around in the bloodstream and goes to all of the cells in the body. And the glucocorticoids are steroid hormones, and the parent molecule that these are synthesized from by the adrenal gland come originally from cholesterol. So it's a fat soluble hormone. It's very amphiphilic. Both it can exist beautifully in water as well as in lipid, and it can scoot right through the cell membrane, which it does, and binds to glucocorticoid receptors on the inside of the cell, in the cytoplasm. And as soon as it does that, it activates the glucocorticoid receptor, which is another interesting molecule in that it's really a transcription factor. So it's a receptor, hormone receptor on the one hand, but it's also a transcription factor because as soon as the hormone binds to it and activates it, undergoes a conformational change that allows it to translocate into the nucleus where it binds to DNA and activates genes. And it was thought at the time, particularly back in 1994, 95, that perhaps what was going on was like AP1, the glucocorticoid receptor translocated to the nucleus was interacting with NFkB in some way, in a protein-protein interaction, and therefore sterically and hindering NFkB to bind to the promoter region of IL-2 and to then activate its transcription. But it didn't really make a lot of sense. And when the people looked for a physical interaction, they could show it. You can, in science, you can find anything as long as you look hard enough. If you know what you're looking for, and you look and look and look, and finally you see it. This is what happened with glucocorticoids receptors. They were found to interact with NFkB, but the experiments that were devised in order to show this were really overexpression of both the glucocorticoid receptor as well as NFkB, and it was really unphysiologic. And Michael Karin felt that it really wasn't probably the way steroid glucocorticoids really worked. It was known by now that for the most part, NFkB, the nuclear factor kappa B, which was originally discovered back in the 80s I'm having a senior moment here. Originally, NFkB was found to be a cellular proto-oncogene, and it was discovered by Howard Temin from one of the avian retroviruses that was an avian leukosis virus for reticuloendotheliosis virus, which would cause sort of a macrophage transformation so that this oncogene, cellular proto-oncogene, was called REL, R-E-L. But other people also found it subsequently and didn't know that, and they named it a nuclear factor of kappa B. So anyway, that's a little bit of the history. It was known that there were at least two proteins that were involved in NFkB, one that had a 50 kD molecular size, whereas the other one had a 65 kD molecular size. The nuclear factor of activated B cells was essentially usually found in the cytoplasm. And if you activated the cells, like say through the T cell receptor, so that you would activate NFkB activity, you would find that NFkB translocated to the nucleus. In addition, it was also found that there was another molecule that was called inhibitor of kappa B that was also present in the cytoplasm. And when you activated the cells, if you decreased the inhibitor of NFkB, then NFkB would translocate to the nucleus and do its deal. So that's sort of what was all known at that particular point in time. And so what Michael Karin decided to do was to really look into this inhibitor of NFkB. And so I'm not going to show you any of the data from this paper because I really labored going through it. The figures are multi-components and very difficult to follow exactly what was all was done. A lot of the materials and methods are in the figure captions. So I recommend that you read both of these papers that are on the website. But I'll tell you what the bottom line is on this paper by Karin. And I liked the paper by Karin much better than I liked the paper by Al Baldwin's group. And Al was from the University of North Carolina. In the discussion to this paper, what Karin says is that collectively, all of the results that he presented define a simple mechanism whereby glucocorticoid repression of NFkB activity occurs. And it's not by the binding of the receptor to NFkB. Rather, it's the induction of transcription of the IkB gene, IkB alpha. And as that then is transcribed and translated, and the IkB is elevated, it prevents NFkB from translocating to the nucleus and becoming active. In addition, the increased concentration of IkB in the cell, in both the cytoplasm as well as the nucleus, when it goes into the nucleus, it sequesters NFkB that's already bound to the DNA and promotes its dissociation from DNA. So, and then causing it to then translocate back into the cytoplasm. And that's how it works. And that's interesting because, as he goes on to point out, this accounts for almost all of the immunosuppressive and anti-inflammatory activity of many, many cytokines, including interleukin-1, interleukin-2, interleukin-3, interleukin-6, interleukin-8, TNF, interferon gamma, GM-CSF, and MHC class 1 and 2 genes. I mean, that's just about the whole ball of wax of immunological molecules that are important for the immune response. In addition, he references other papers where it's been shown that glucocorticoids in vivo inhibit the in vivo, if you administer anti-CD3 to mice, the glucocorticoids will inhibit the production of IL-2, IL-6, interferon gamma, and TNF in vivo. So basically, this is why the administration of glucocorticoids have become so important in the clinic to use, particularly in inflammatory situations, when you really need so-called big guns. And of course, we're in the process of the COVID pandemic right now. And one of the very first drugs that was found to be effective, if given early enough, were glucocorticoids, dexamethasone, which is a synthetic glucocorticoid. And of course, that's what the former president was given when he was rushed to the hospital, Walter Reed in Washington, when he came down with COVID symptoms and his oxygen saturation started falling. And they started really worried that he was going to rapidly go downhill and end up on a ventilator in the hospital, which he would have had they not given him glucocorticoids as soon as they saw that the oxygen saturation fall. And just to put a plug in to this whole area, one of the things that's happened in the whole COVID pandemic is that the doctors that have been taking care of most of these COVID patients in the ICU have come from a pulmonary critical care anesthesiologist background. They're used to dealing with heart failure patients that run into pulmonary edema and problems, and they knew how to put patients on ventilators, and so that's what they've done. But really, what needs to be done in these patients is to block that inflammatory response as soon as just like they did with the former president, as soon as you think that you're going to be getting into trouble. And so one more point I'd like to make with regard to that is that it's the signs and symptoms of disease in COVID caused by the coronavirus are not due to the presence of the virus. They're due to the inflammatory response to the virus, the immune inflammatory response to the virus, which are caused by the pro-inflammatory cytokines, interleukin-1, interleukin-6, TNF alpha, interferon gamma, GM-CSF. All of these cytokines are activating the inflammatory response that causes the vascular leak that's responsible for the leakage in the lungs that fill up the tiny little air spaces and lead to respiratory, basically insufficiency. So I think that what we really need to do in immunology and modern day immunology is to create a whole new subspecialty of medicine called medical immunology, sort of as a takeoff on medical oncology. We need people that understand the immune system and the inflammatory system and all the molecules involved, and they should be taking care of the patients when they come to the hospital with these very serious inflammatory diseases. So I'll get off my soapbox and I'll stop there. So don't forget to like the video, click on the bell, check out the website. And there's one other thing I want to show you is you can pick up a lot of this business in my book, Molecular Immunity. I was going to have this ready, but I forgot. So check this out. You can learn a lot there. I'll stop. Thank you for listening, and I'll see you next week.